Hello, everyone. My name is Natasha Sheybani, and I'm a postdoctoral fellow in oncology at Stanford University. I also serve as the senior scientist at the Focused Ultrasound Foundation, where part of my role entails directing the gene therapy program. And so to prepare us for what I hope will be a fruitful discussion at this year's GBM workshop, I'll be giving a brief overview of the state of the field for FUS and gene therapy. To begin, I wanted to highlight the degree to which gene therapy is flourishing in cancer. The pie chart to the left here depicts data collected up to 2019, and I hope this emphasizes to the audience that cancer really comprises about 65% of the indications addressed by gene therapy clinical trials. There are a multitude of approaches to gene therapy, and I'm highlighting several broad categories here on the right. We have gene replacement, where the goal is to replace a dysfunctional mutant gene with a healthy gene. Gene silencing, which entails knocking out or inactivating an inappropriately functioning mutant gene. Gene addition, wherein the goal is to introduce and usually overexpress new genes into cells as a measure of disease protection. And lastly, we have the ever-burgeoning category of gene editing, which can enable the precise and permanent manipulation of genes at specific locations in the patient's genome through tools like CRISPR-Cas9. In the context of GBM, there are a variety of strategies that we have at our disposal for deploying genetically based therapies. And I offer some examples of in vivo gene transfer approaches here on the left, along with their advantages and limitations. For the sake of time, I'll just highlight what they are. They include suicide gene and prodrug therapy, oncolytic viral therapy, immunomodulatory therapy, as well as deposition of genetic materials using synthetic non-viral vectors. And on the right-hand side of this slide, I highlight an example of ex vivo gene transfer through that of chimeric antigen receptor or CAR T-cell therapy. As illustrated by this workflow, we have peripheral T cells that are sampled from the patient's blood, following which they're genetically modified to produce chimeric antigen receptors. And after this, they're infused back into the patient in order to surveil for and attack tumor cells expressing the antigen that they're programmed against. And I'll briefly touch on some of these gene therapy strategies in greater detail over the coming slides. With all of that said, there are several challenges that remain for gene therapy, and I'm summarizing some of them on this slide. Most importantly, there remain critical barriers to achieving localized, minimally invasive targeting of gene products, as well as improving transfection and transduction efficiency. And herein lies, I think, a tremendous opportunity to leverage focused ultrasound to ultimately improve the deployment of gene therapies. And to that end, I'd like to spend the rest of this talk discussing the role of FUS in advancing gene therapy. Over on the left, I'm highlighting some mechanisms of action of focused ultrasound that bear relevance to gene therapy applications. And you can see they're quite diverse in nature. But the thing I want to emphasize is that across the preclinical literature, we're already coming to find that multiple of these mechanisms of FUS can aid in one, improving gene vector delivery and transfection or transduction efficiency. Two, potentiating gene editing techniques. And three, enabling remote spatially targeted activation of gene products or control of genetic circuits. And in the next few slides, I'd like to just touch on some examples of each of these mechanisms, beginning with the first strategy highlighted here. And before I do that, I think it's important to note that, of course, a majority of CNS-directed drug and gene therapy applications, including those in GBM, are hindered by the presence of the blood-brain and blood-tumor barriers, which I'll refer to as the BBB and BTB respectively. So gene therapy, first and foremost, stands to vastly benefit from the non-ionizing, non-invasive mechanism of FUS-mediated barrier opening, whereby we can safely and transiently disrupt the BBB and BTB by applying a localized acoustic field in the presence of systemically circulating microbubbles. And of course, this will be a topic of tremendous interest to the discussion over the 2 days of this workshop. So there are now myriad preclinical publications that have demonstrated the ability of FUS-BBB opening to enhance delivery of either viral or non-viral vectors to the brain. This has sort of ranged across both the naive brain setting as well as a variety of different pathological settings. But just for the sake of time, I wanted to highlight here this example from Richard Price's group at UVA, which has demonstrated a proof of concept for non-viral vector delivery in GBM. As you can see, mice-bearing U87 glioblastoma tumors underwent BBB and BTB disruption with FUS. And this was, of course, done in the presence of systemically circulating brain-penetrating nanoparticles that were bearing a reporter gene. In this case, they saw improved transgene expression after just one round of barrier opening, and this was conserved across 2 different peak negative pressures of FUS that they evaluated. And the representative IF images on the bottom right here show that compared to systemically administered BPNs alone, focused ultrasound mediated BPN delivery actually improved the penetration of these particles well beyond the confines of the tumor microvessels and into the tissue itself. And while I don't have a whole lot of time to touch on them here today, recent years have seen the publication of a variety of other non-viral approaches for gene delivery to GBM as well, most notably with cationic microbubbles or microbubble-like liposome complexes. So I'm offering some of that literature here for those who are interested in delving a little bit deeper into those platforms. Another area where BBB opening and potentially other mechanisms of FUS are poised to play a role is in gene editing. And the groundwork is already being laid for establishing the role of FUS in efficient and targeted delivery of gene editing systems like CRISPR-Cas9. So this study I'm highlighting here was published earlier this year and utilized lipid polymer hybrid nanoparticles to refine the delivery of CRISPR-Cas9 plasmids targeting MGMT, which is a drug resistance gene to temozolomide. And in an orthotopic GBM model, these particles in combination with FUS-BBB opening substantially improve delivery and transfection efficiency over their control and monotherapy counterparts. And I'm sort of highlighting those areas of data with these the blue box and arrows. And you can also see that this extended to improved survival in the mice that they treated with that combination as well. And finally, there's a fascinating category of emerging applications where other mechanisms of FUS are being leveraged to remotely control gene products or activate gene circuits in a spatially controlled manner. For example, this group of studies has demonstrated the feasibility of leveraging ultrasonic heating to achieve spatio-temporal control of gene expression in different tissue settings. And for those who are interested, I think, some of the fascinating implications of this approach for cancer treatment were highlighted quite nicely in a CCR focus piece by Krit Moonen. In a similar vein, there are some other tools coming down the preclinical pipeline that capitalize on the remote capabilities of FUS, as well as the deep tissue penetration of sound waves. And this combination is sort of useful in helping us yield tools for gene delivery in optogenetics, in acoustogenetic control of CAR T cells via ultrasound controllable genetic circuits, as well as acoustic transfection as an emerging alternative to traditional sonoporation for delivery of systems like CRISPR-Cas9. And again, I'm citing some of the relevant literature here for those who are interested in understanding these tools at a deeper level. And in addition to the strategies I've already discussed, I want to, of course, point out that there are some additional strategies that underscore a rich interface of FUS-mediated gene therapy with immuno-oncology. So there are a variety of ways that we could envision using gene therapy approaches to potentiate immunotherapies. And the 2 strategies that I haven't really talked about yet in this regard are improvement of CAR T cell delivery and delivery of other adoptive cell therapies, as well as potentiation of tumor vaccination strategies. So first, on the topic of CAR T cell improvement, there really hasn't been a lot of work published explicitly on the combination of FUS and CAR T in GBM just yet, but it's worth pointing out that recent work has established the ability of FUS-BBB opening to enhance cell delivery in a tumor model. So the work that I'm highlighting here is from Kalervo Hinninen's group, demonstrating the delivery of HER-specific natural killer cells in an orthotopic HER2-amplified rodent brain tumor model that was meant to recapitulate aspects of metastasis of human breast cancer. So the accumulation of these NK cells was quantifiably improved by delivery with FUS-BBB opening. And as you can see, this translated to both growth restriction as well as survival benefit in the animals as well. And I want to point out also that there's a growing body of literature establishing the immunomodulatory effects of FUS on cytokine expression, activation of the vascular endothelium, antigen liberation, as well as phenotype and function of immune cells that are either local or peripheral to the tumor microenvironment. And while I'm not really getting to highlight that body of literature here, I point out its existence in order to emphasize to those unfamiliar with FUS that there's a great deal of related work underscoring our hypotheses for how FUS can improve not only the delivery, but perhaps the persistence of genetically engineered cell products in GBM. Finally, there are several preclinical studies that have demonstrated a role for FUS in delivering and/or activating agents like microbubbles or nanocarriers that bear antigen encoding or immunomodulatory genes as yet another strategy for addressing bottlenecks in the cancer immunotherapy cycle and/or enhancing the performance of immunotherapies like checkpoint inhibitors. And so I'll leave you with this one final example from Kathy Ferrara's group at Stanford, which demonstrates the feasibility of FUS-mediated cytokine transfection for potentiating PD-1 blockade. Here they implement a paradigm combining low-frequency ultrasound with tumor-targeted microbubbles that were designed to partially destroy tumors and then transfect remaining viable tumor and stromal cells with plasmid DNA encoding interferon beta. So this combinatorial therapy was meant to trigger an immune response that cooperates with immunotherapy. And their group shows that it indeed harbors superlative efficacy in recruiting macrophages and CD8 T cells, as well as exerting tumor control in conjunction with PD-1 blockade. So while this study isn't in a brain tumor model, I think it again highlights yet another interesting opportunity for being able to sort of kickstart the immune response to GBMs going forward. So to close, I hope this brief presentation has impressed upon this audience that there's really a wealth of preclinical activity being dedicated to expanding the role of FUS in gene therapies. And these all, in one way or another, hold promise for GBM. While I exclusively highlighted cancer applications today, there are applications in neurodegenerative diseases as well as other pathologies that could potentially unveil important discoveries or principles for GBM applications going forward. And of course, I hope that some of the discussion at this meeting will help us map out the role of larger animal models in GBM gene therapy, as well as what the most promising approaches are to invest in within this sort of armamentarium of approaches that we have at our disposal. While there aren't any active clinical trials evaluating FUS-mediated gene therapy in GBM just yet, we also anticipate that these will emerge from the pipeline as a result of the forthcoming discussions. So with that, I'd like to thank you all for your attention and welcome you to contact me with any questions or requests for more information. If you'd like a deeper general overview of FUS and gene therapy, you can also check out the Foundation's recent webinar on this topic, which is located on their YouTube channel. Thank you.